So my name is Veli Vural Uslu. I used to be at EMBL between 2009 and 2015, first as a PhD student and then as a bridging postdoc. And what I'm doing now is I'm a research group leader in a plant research institute, Agroscience Research Institute. And what we are doing is more in terms of understanding the plant-environment interaction. So we are digging into the basic biology, so how plants respond to the environment and change their developmental trajectory according to the environment. But we are also trying to use methods of manipulating it. And one method basically is RNA delivery that turns out to have very important applications in agriculture. So one part of my lab is more into the basic science of it, and the other part of the lab is more on the applied part of plant biology.
Okay, so science and technology. So let's start a bit from the beginning. And what did you find challenging in the project? What did you find particularly satisfying? How did your PhD go?
It went really down at the beginning. Okay. I didn't know what to do because developmental biology was new. And it was a time before CRISPR, so things were actually very difficult if you are actually particularly doing genome biology as a part of developmental biology. Because what we were trying to do was trying to understand how the genome is organized in mammalian systems so that there is very specific gene expression. So for us, it was a particular gene called MYC, but it had to be expressed pretty much everywhere, but how much it's going to be expressed is a question. And the puzzle that Francois introduced at that time, this was a gene, a single protein coding gene in a 3 megabase window, which is 1/4 of yeast genome, and there's a single gene. So how is it possible that these regulatory elements work in such a coordination so that we get a precise MYC expression? So to understand this, we had to make mutations, but it was a gigantic region, and we really didn't know how to tackle with this problem at that moment.
So a tiny gene and a lot of regulation, exactly. A lot of what people would call junk DNA, long time ago to figure out. And what did you actually find?
So what we did is Francois came here with a particular method called targeted meiotic recombination. He's a French guy, so he was always calling it TAMER. For a shorter version of it. And what we did, basically, we deleted these sequences far away from the gene itself. And we looked at, not only deleted, but for example, we did duplications. We put these regulatory sequences far away. We put them closer to the gene. We inverted these regulatory sequences. And then we checked basically what is taking place in terms of gene expression, and then further on in terms of developmental phenotypes. So this was the main question, and what we found is there are elements far away from MYC itself that is extremely important for very specific tissues, for example, the face tissue. And if you make deletions or if you perturb this regulatory interaction, you could actually increase the susceptibility to get certain cancers like acute myeloid leukemia, or cleft lip palates, like this developmental phenotype that is actually rather common. But these things were these regulatory elements are not like gene deletions. You delete a gene, you get a phenotype. It's not like this, but you actually increase the susceptibility. So whenever there are some environmental factors playing a role, then actually you start seeing the phenotype. So this is why it was the genetics of susceptibility, in a way, what we were dealing with.
I mean, what I find fascinating of what you are saying is that it seems like the technique were mostly traditional molecular biology, where you're mutating or deleting or taking out pieces or swapping pieces. But then it turns, from what you say, it turns out that it's more like I would say, architectural, problem, right?
Precisely.
So, you get the architecture of the whole gene environment dictating the expression of the gene, which is a rather, I guess, novel and hot, I would say, scientific concept, right?
Correct. And I think that what made it very challenging is exactly this architecture, because what people did before is they deleted the gene, they overexpressed the gene, and that was it. But for us, it was so many regulatory elements all around the place. We basically, in my PhD time, only me in the lab and many other people were working. We had more than 80 deletions, duplications, inversions, and these were all in vivo mouse lines that we had to keep. And this was an enormous work, enormous library of deletions that we had to bring all together to make an interpretation out of this. What is this architecture about? So that's why I think this how we could actually publish it in Nature Genetics, Nature in respected journals, and this is why I think it also got a lot of citations since then.
Yeah, no, it's cool. I mean, I find really fascinating because I mean we know that in biology genes are used over and over in different contexts, but it's very difficult to actually understand the tight regulation by which in one set of cells the same gene is doing a certain thing and in another set of cells is doing another type of thing. So I think, you know, I can relate a lot. We do a lot of things with another gene that is very important in development, which is called Notch, and Notch has the same problem, right? You know, it's deployed many times in different contexts, and people have been going crazy for a long time trying to understand why it does X here and epsilon in the other set of things. So it's nice that you also were able to relate it to, you know, disease.
Yes.
And actually, I was reading from your CV that when you moved for a postdoc, you actually were moving in somewhat related field initially, right?
Yes, so it was, what I wanted to understand was how this environment affects the susceptibility, but mammalian systems are way too stable to be able to understand. And then actually I got to meet Rüdiger Hell from the university, and then he introduced this concept in plants, which was quite strange to me at that moment. But then it clicked immediately, because what he was explaining was that the plants also show developmental differences, like it happens in some diseases and so on. And what happens is, if you think about a plant that, you know, just the grass that is growing in the pavement, basically it snows on them, they are fine, it shines 45-degree sunlight on it, it's fine, it is flooded, it's fine. So how is it possible that the plants can adapt to this environment? How this environment affects the gene expression in plants so that they manage to survive or they adapt themselves? And I thought there was a lot of space that I could explore by, you know, taking the plant and then treating it in a different environment to look at the different developmental phenotypes of the plant. So this was this rather smooth transition, but of course using a completely different organism, in this case, plant.
It's amazing. I mean, they don't even have a nervous system, so
exactly.
They have to do that without a nervous system. But okay, so then you moved from animals to plant at that step, essentially.
Exactly.
Okay,
so this was the main question.
But was there anything, usually when you move, you know, PhD to postdoc, you try to keep something at least related, or maybe technically or scientifically. So you changed completely, or you kept some relationship with what you have done before?
I thought I actually changed completely.
Okay.
I thought now I'm working with plants. And this was, I was really looking for a place that I could actually make my own niche, not follow something that Francois was doing, just trying to make something better, more than what he was doing. So it's not like Olympics, but it's just like trying to define my own niche. So that's why I thought it's plants, and now I move into a completely different direction. But what it turned out that actually the regulators of these environmental sensors and the developmental regulators in plants turn out to be pretty much the same things actually that we were dealing with in mice, which is the polycomb proteins, H3K27, histone methylations. These are all the things that turn out to be very familiar to me from my PhD work, but I never expected that I was going to encounter with them when I actually started working with plants.
Okay. So you were able, in other words, to assign a function that was very well studied in an animal cell to a plant cell.
Precisely.
Okay. So that's interesting because, of course, you know, we know that the plant field for some aspects is a little bit behind the animal field. I mean, you would say that except for some fields or subfields, the plant biology is a little bit catching up. So that's quite interesting. But what was your feeling going from, in fact, you know, I say this as an animal biologist. I would be lost if I had to get into, you know, the literature of plant science. You know, it's almost like going back to school, right?
Absolutely.
Yeah. How did you feel?
I felt at the beginning I was very excited. So I think I didn't really understand what I was dealing with. But then I had to catch up with the knowledge, and of course I had to build up something on top of it. And I had really difficult time basically at the beginning of my postdoc because there was not so much of, I would say, good quality data coming out of my research. And I also found it fascinating. I think it is exactly as you described. Plant field is moving like a few steps behind the animal field, so that the methods are way behind. It's very difficult to apply them. There are not so many people who work on this so that you could actually immediately start a network to develop the methods rather fast. So I found it very difficult, but I was very lucky actually that I had Rüdiger Hell as my boss, who supported me a lot. Even at the time, you know, things were not really running well. He was not really getting the results that he wanted or he would love, but he really supported me, and I think it was rather a long transition. So I cannot say that I was very successful at this transition phase, but thanks to his patience, things started coming out basically after the second year of my postdoc.
Okay. Well, I guess, you know, changing so radically, there's a learning curve that takes a bit of time. Yeah, no, and the other fascinating thing about it is that you refer to polycomb, but then I read in your CV also TOR pathway. These are things that are very, very well studied in animals. You would think that, yeah, they basically, you know, if we think about polycombs, they regulate gene expression. If we think about TOR, they sense nutrient metabolism. So would you say that in the end they're doing the very same thing in plants as they do in animals, or there are also differences?
I think you referred to this point earlier with referring to the central nervous system when we were talking about the neuroscience part of it. I think, so what we found is that there is the environmental sensor, TOR, and so this became the cover story of Journal of Experimental Botany in March this year. What we found is that TOR is acting on the polycombs directly, and also Jan Shin's lab from Harvard showed it basically in a recent publication, and this is regulating the H3K2 and 7 deposition in certain or critical genes basically at the promoters. So that's why we think that it is sensing the environment and it is acting on the genes that are important for development. But the thing is, we already knew the polycombs were actually important as a developmental regulator in plants. For flower development, it was important. For lateral root development, it was important. But the link between the environment and the developmental trajectory was rather missing. The link was missing there. And what happened in animals is we know that TOR is there, and there is hardly any link between TOR and the polycombs because our body is rather fixed. So we don't have something called developmental plasticity. For example, in plants, the roots go underground, and then when they appear, you see that the leaves start appearing from there. So they are very plastic in terms of their development, which is not the case for mammalian systems, or most likely the systems that can run away from danger where there's a central nervous system, so that they don't have to adapt the environment. They can actually react to the environment by moving around. So we think that this is actually very critical in terms of this separation, that polycombs are regulating the developmental trajectory only, or sensing the environment and regulating the trajectory of development in certain plants.
Okay, so that's a very interesting lesson to think about. The other thing that struck me from your research story is that you seem to be getting more and more also into techniques and biotechnology. You were referring to in your CV about new methods that you're using to act on plants. So can you tell us a little bit about the technical part of your work and what you're developing now?
Yeah, so what I knew from this polycomb regulation, environmental sensing and so on, I thought, well, we can learn this, but we really need to change it. And the way that you could possibly change the polycomb deposition or the landscape is actually introducing RNAs, which is very, very difficult in plant biology, especially if you would like to do it outside in the nature, where you do not want to go through a completely new plant line, and you don't want to go into plant breeding, which takes years. So we really needed simple methods basically to be able to modify this. And one very simple method is RNA delivery. So the problem is RNA delivery was thought to be very easy in plants, and the early research didn't turn out to be actually correct. So that's why we thought it would be very easy, and it turned out to be a very difficult problem to be able to get these RNAs in the cells. And then one of my colleagues, Thanasis, so he managed to develop a method called high-pressure spraying technique to be able to deliver these RNAs, double-strand RNAs, single-strand RNAs, into the plant cells directly. And then he triggered an RNAi response, or that's basically something that comes along with this processing of these single-strand RNAs, double-strand RNAs in the cells. So we thought, well, actually, this could be a very interesting way of using in certain applications. We couldn't directly use it for the polycombs, but we thought that actually there could be very interesting applications of it in terms of RNAi trigger against plant pesticides. Mm-hmm. So this is how a simple method basically we thought we could use turned out to be very interesting, turned out to have a very interesting biotechnological aspect in plant defense and agriculture.
Okay, so it sounds like you were thinking about developing this for fundamental biology, but then you realized it was more appropriate for applied biology.
Precisely.
Okay, so why is that? Why it didn't work for fundamental biology and it became useful for applied biology?
I think the problem was that the efficiency is not, it was below certain threshold for applying it directly for basic biology. But RNAi machinery in plants particularly work with a positive feedback mechanism, because there is an enzyme particularly called RNA-dependent RNA polymerase, which mammalians actually lack. So this is, since it is RNA-dependent RNA polymerase, it actually amplifies the RNAs that we sort of introduce into the cells. And this is how we could trigger a certain response, even if we deliver certain very, let's say, little amount of RNA. This is enough for us to get the response. So that's why, because of this efficiency difference, it turned out to be actually very interesting to use it for in the context of RNA interference.
Mm-hmm. Mm-hmm. So and I guess the thing I did not understand is how does it work? I mean, how do you transfer the RNAs into the plant cells that has a cell wall on top of a membrane? Like animal cells will have just the membrane, but the plant cell, of course, have the plant skeleton, which is the cell wall, right?
Yeah, so what my colleagues did at the time is they actually got an airbrush. So this is an airbrush that people use for painting the cakes, or you see actually in Heidelberg under the bridge, people make graffitis out of it. So it's just very a simple airbrush that people use with a compressor. But instead of putting paint in the reservoir, we put RNAs. And then we directly spray these RNAs onto the meristem and also onto the leaves. Okay. And this is actually enough, this pressure is enough to deliver the RNAs without killing the stem cells or the leaf tissue, for example. So that's why it turned out to be actually a very useful method that you can even take it to the field, just taking the compressor and the airbrush with you, and then you can actually deliver any kind of RNA.
Okay. So it's like you're almost punching a hole in the cell.
Exactly.
And then deliver the RNA particle, and then the hole somehow gets sealed or whatever.
Exactly.
So
and even if it's a leaf surface, basically you can cover the leaf surface as little as 25 microliter material, which is almost nothing, and then you manage to deliver basically it to the whole.
That's great. So science and technology, and I read something about sulfur sensors and genetics as well. So this is another technology project.
This is another technology project. So this is actually, we thought, as I mentioned before, I never thought that this would be a genetic regulation, but I thought it would be a very much of a metabolic regulation between the environment and the developmental trajectory. So that's why we thought maybe we could actually develop some sensors, in vitro sensors, to understand how the environment, particularly the nutrients, is acting into the intracellular nutrient levels, and then maybe they act on the gene expression. But after almost 2 years of research, actually, we couldn't develop the sensor. So that's why we had to leave the project aside. We thought that, well, these are not the proteins that we could use at the moment. I thought it could have a very high impact basically on the field, but sometimes science is like this. Failure is failure. We learned, what we should not do, what we should be looking at. So that's why it was actually a lot of effort that was just put aside. And this is something that I actually really like to highlight, because also in my PhD or in my even today, most of the experiments actually, or most of the hypotheses that we come up with, they fail, but we always cherry pick this 10% very interesting stuff that go into publications and then we highlight them. But I also really like to explain that we are not special people that is doing crazy all the time. We are fantastic scientists, but actually we are just like normal people. We fail a lot, but when we get something very meaningful. So that's not something that that people should think scientists as very special people, but we are just like people actually. So that's why I really like to highlight these failures as well when it comes to the postdoc life.
I think it's interesting, and certainly, of course, like in other fields, these failures are teaching something, right? So, maybe it's not something that you end up publishing, but it's something that you can learn from to just, design a better experiment the next time, or focus more on a field that is more promising. So I guess, it's still time that is well invested, although, certainly, we know that in this job there's a lot of frustration as well, and part of the success is actually being able to restart and sustain this level of frustration sometime.
And people sometimes ask, what changed between a PhD time and a postdoc time? I think it was very difficult for me to recover from my mistakes, recover from my failures in my PhD time. It would take like a week of, almost a therapy for me to get back to the lab and work hard. But in postdoc time, I actually got to understand this is really a part of our life, that we just sometimes fail, we move on to another thing. And I really like this idea of recovering fast by experience from our failures.
Yeah, so I guess it's part of the training, you could say that.
Absolutely.
Okay. Of course, another thing that really comes out of your very impressive CV is the science dissemination that you have been doing very early on, I have to say. You started quite early during your training to think about how to communicate science, get involved with your local community, I guess. So where does that come from? And tell us a little bit about how it developed, maybe before EMBL, at EMBL, and how is it going now?
So I think the quick answer is it actually started before EMBL, but I never thought that it was something that I could take outside of EMBL. It was EMBL Theatre Club, we really enjoyed it. EMBL Photography Club, we really enjoyed it, and it was there. But what happened is after EMBL, I decided to stay here in Heidelberg, and everybody from my PhD year actually left. So it's like almost 40 people that we got to see each other almost weekly or every day in the corridors, and they were not there anymore. And I had to start to a new subject, and I didn't know anybody around. So this is how I thought, well, maybe I could make some friends with theater. So I started basically putting out posters in the campus, and people basically came. It was a group of 10 people initially who wanted to try out theater without any theater experience. And then what I realized, actually, it was for theater, but it turned out to be like almost a therapy session, because when PhD students come together, they are directly from their lab, they are very frustrated with their failures, with their interaction with their PIs, with their legal status, with visa issues and so on. And it turns out that the PhD students had a lot of extreme emotions, either extreme joy because they found something, or extreme anger because something didn't work, or extreme anxiety. But when you look at them basically in their daily life, scientists hardly show any emotions. And it's a job, it's almost like doing extreme sports, but just not talking to anybody about it. So that's why I think it gave the PhD students a very good platform for being on the stage, trying to explain what they think, what they have. And of course, they are very much into science. So this sort of laid the ground of getting a lot of people, or forming a community that wants to express themselves. And these things are generally science related. so that we actually could find this crowd to bring science out of the lab to people with more emotions and more, let's say, in a more friendly and fun way.
Well, that's good. So you think that, I mean, from the way you describe it, it seems like that you think that the emotional part is the part that resonates with people that are non-scientists. Is that so? I
completely agree with this statement. I think what is really lacking is that we are presenting very cold-hearted data. It is the data, it is there, you cannot really bend it here and there. But the way that we perceive it is we get actually excited about it, or we are not happy with this data. So we really change a lot and we really block it. Whenever it comes to explaining some sort of scientific fact, we really stick to the facts, we completely isolate ourselves from it. But actually what people want to listen and how they get more engaged into the subjects is with the emotions, with the story, how did it came to this point, how we feel about it, what it means for them, how do we feel about the people who are listening to it. So this is the part that I think it was lacking in science, in terms of science communication. And this theater experience sort of revealed why putting the emotions or giving some space for the emotions when we are presenting scientific data is actually important for the community.
I guess it's also positive emotions, right? I mean, do you think that being excited about what you're doing contributes to explain it better or to make it, more valuable in the face of the public listening? I mean, is that so?
I think it is. I think what is very important is that trying to engage people into some sort of conversation. And when you show the numbers directly, people do not want to get into the conversation. So emotions, I don't think that it may have, but I'm not sure if it really has an importance of telling things better or something. But it definitely has an importance of engaging more people into the conversation, into your conversation, into the things that you want to say. So that makes it much easier for us to communicate science. So it lays the ground basically for communicating further science, just bringing these emotions out. And as you said, not only this anger, fear, like negative ones, but also the joy that we find something. It's just you see one bend, and then you get like, this is the day of my life. And then it's just a Western blot band. And if you just show the Western blot band, you cannot really get people into conversation. But if you say that, well, I'm going to tell you the best day of my life, and this is this tiny little band. And then people get into the conversation.
Sure. And then you have the chance to also explain what is that tiny little band.
That's the idea.